Thank you very much, everybody, for joining us today at this session, which promises to be very exciting. So I'm Olga Vitek. I'm representing the Virtual Media Outreach Committee of US HUPO. And the committee works very hard trying to connect all the members of our community in these difficult pandemic times. And so today I am really excited to welcome you to the session, which will be hosted by Dr. Ryan Kelly. So Ryan is an associate professor in the Department of Chemistry and Biochemistry at Brigham Young University. And prior to this position, he spent 13 years at Pacific Northwest National Laboratory. And during his career, Ryan has led the development of many technological solutions for ultra-sensitive biochemical analysis. And our committee felt that Ryan was the perfect person to host the committee of experts who will speak today. So welcome, Ryan, and thank you for being part of this effort.
Thank you very much. Okay, let me go ahead and share screen. So we are excited to have 6 experts plus me serving as panelists here. And basically we're going to have each panelist spend about 3 minutes introducing themselves and providing just a little bit of their perspective with a couple of slides. And that will take about the first half hour, I think, by the time we get through everybody, and then we will open up the Q&A portion of this webinar. And so make sure you enter your questions in the chat, and we'll go from there, starting with, and we're going to start off with Jenny Lill, who can introduce herself and provide her flash talk, if you will.
Great, thank you, Ryan and Olga. Thanks for the invitation to be part of this. So, for those of you that don't know me, I'm Jenny Lill, and I run the NGS and Proteomics group at Genentech. Next slide, fine. Thanks, Ryan. So we do next-gen sequencing. We have a translational mass spectrometry department that's headed by Wendy Sandoval. We're going to hear from Chris Rose, who's the director of the Discovery Proteomics department. And then we have our informatics group that's headed by Corey Bakalarski. And with these different teams, I think we're uniquely positioned to answer lots of questions in the omics space. Next slide, please, Ryan. So about 1.5 years ago, we asked the exact question that we're tackling today. What is the future of proteomics? And we were really specifically asking this for industry. So we interviewed many of our key stakeholders and collaborators, our consultants, our friends in the field, and we generated this word cloud based on the responses that we got. And you can see that there's lots of different things here that we're going to be hearing about from all of the panelists today. But at Genentech, because we wanted to focus on a few key areas, we decided that our main investments for the next couple of years are to work on single-cell proteomics, to improve our chemoproteomics suite of tools, and also to build on our interactomics tools as well, so that we could answer all the different biological questions that are coming along. Next slide, please. So Chris will tell you about the single cell proteomics side of things, but what I was particularly interested in sharing with you today is on the interactomics side of things, we have a number of different tools that we're building or bringing in to look at intracellular protein interactions, particularly nowadays trying to look at transient interactions, low-level material, and be able to look at these in primary cell lines to really capture the biology, especially when it's in a transient state. But what we realized from our conversations with people is that there's still a hole in terms of looking at membrane proteins and looking at the receptomics side of things. So we have a team headed by Shanghai Kao who has a number of different tools that are complementary to mass spectrometry. That's what I wanted to highlight today is we have these different screens based on a number of libraries, so single transmembrane, GPCRs, various different membrane-bound proteins that allow us to do these screening to look for interactions in that space. And so we can pick this up when we come to the discussion later, but this is the area that I wanted to highlight as a growing need within the proteomics arena. And with that, I will hand over to Chris Rose.
Thank you very much, Jenny. So yes, I'm Chris Rose. I'm the Director of Discovery Proteomics at Genentech. And there we go. I can start my video. A couple of the areas, as Jenny mentioned, that we're really interested looking forward, and partly where my group has been interested in the past few years, of course, is single cell proteomics, where we've done a lot of work trying to understand kind of how to approach it from a quantitative aspect and how do you design your experiments. But as we go forward, I think a lot of what we see is going to be changing is obviously some of the automation and low-level sample preparation. We've already been seeing a lot of this with some of the more commercialized versions of things that Ryan has actually worked on in the past. Beyond that, I'm very interested to see how instrument innovation occurs. You know, I think it's really nice nowadays that we actually have a little bit of competition between a couple of the big mass spec vendors. And I'm hoping that that's just going to drive innovation, potentially more sensitivity, and allow us to analyze some of those samples that we are trying to go at lower levels. Beyond that, I'm particularly interested to see how intelligent data acquisition evolves. And I think there's already been some really great examples of this with MaxQuant Live, with some of the work in Orbiter that Devin Schweppy's lab has been working on, as well as work that we've done internally. I think that these types of tools can hopefully enable us to do new types of experiments or allow us to do more advanced analysis of, say, some of these lower level samples where we might have to do more intelligent acquisition to actually get the depth that we would like to see if we're trying to reach the types of proteins that we want to monitor when we're trying to characterize a system in therapeutic development. I think today we might actually spend a lot of time, it seems from other slides, talking about single molecule sequencing. But this is one area we're also interested in. Of course, the promises that the different technologies potentially have in terms of being able to identify single molecules, single protein or peptide molecules within a complex mixture, they can have a lot of impact in terms of how we develop therapeutics, how we analyze biological samples. And some of that will depend on how far these technologies go. And I think over the next 5 years or so, we should be able to see how these develop and where we can use them and how they can complement some of our proteomics techniques. And then lastly, the chemoproteomics angle. So there's been a lot of excitement around different types of, say, molecules, small molecules that we can use, particularly covalent modifiers, which have entered the clinic really for the first time. I think we have the first approval of one of those out in the space and therapeutic space. And how we go from, say, cysteine covalent inhibitors into different amino acids and how we build up the databases that tell us which residues within the proteome might be ligandable by these, say, covalent modifiers. I think that's going to be a large focus within the therapeutic development space. And then how do we leverage these chemoproteomic technologies as we go to different types of small molecules or different types of molecules in general for therapeutics. So I think that's another aspect that to keep your eye on, because I think there'll be a lot of development in that space, particularly as it pertains to drug development. And then I think next is Javier, who will be talking.
Hello. So my name is Javier Alfaro. Next slide, please. Right. I'm here I'm a computational mass spectrometrist, but I've taken a keen interest in the single molecule protein sequencing world. And I just wanted to introduce the group to this world. And I came to know it through 2 conferences, but it is much larger than these 2 conferences. One took place in 2017 in Delft, and the second one took place in Jerusalem in 2019. And it's sort of starting a movement of single molecule protein sequencing technologies that are starting to evolve and gain traction. Next slide, please. And so, simply put, this is a collection of physicists and biophysicists that are working towards highly sensitive protein sequencing technologies that are complementary to mass spectrometry. And the dream is to be able to take cell lysates or sample preparations of proteins, prepare them for sequencing, pass them through a single molecule protein sequencer, and then identify the proteins within that sample. Of course, these technologies are very early on. Most of them are proofs of concepts, and they include new breakthroughs in mass spectrometry, high throughput Edman degradation, biological and synthetic nanopores, and DNA nanotechnologies. They tend to sequence peptides using one of three major strategies. One is a fingerprinting approach, which involves chemically labeling specific amino acids, and then sequencing the peptides. And then also there are attempts to sequence every single amino acid in the protein, say using a nanopore. And also there's a community of biophysicists that are maybe not so interested in high throughput sequencing. They're more interested in analyzing protein dynamics through structural analysis. Next slide, please. So this is an area that is gaining a lot of momentum right now, and there's a tremendous amount of investment going into it. You can see that there's investment into companies like Quantum C, Arison, and Nautilus from tech giants, so Amazon, Alphabet, and Google. Next slide. And so I guess my opinion is that the future of proteomics is really bright. We're at a crossroads right now, and we have all of these interesting orthogonal technologies that are coming up. We're not alone as mass spectrometrists anymore. This is a growing movement of new technologies, and how it'll impact proteomics is unproven and unclear. And it's likely that the next 20 years, 10 to 20 years ahead of us will involve merging these technologies with mass spectrometry. What I want to focus in today's meeting is to really have this meeting be about defining the gaps in proteomics accessibly to a broad audience, because a lot of these technologists, they're not mass spectrometrists. So we need to communicate what are the problems that we have to the mass spectrometry so that they can work on various solutions. And yeah, I think that together we can make the future of proteomics happen. And so I'll, at this point, leave it to the next speaker.
Yeah, hello. I'm Magnus Palmblad. I'm at the Leiden University Medical Center. This one slide here is showing some snapshots, if you like, of my journey through proteomics in the last 20 years. But I will talk about where it took me or where I am right now. So I started doing computational proteomics a little bit over 20 years ago, with trying to improve the way we analyzed and identified proteins, bringing in things like retention time prediction, and also trying to get more information, more multiplexing by mixing different isotopic labels. These are stuff that I think most of you have done at some point. So varying the number of nitrogen 15s in plants, for example, doing plant proteomics. Then getting more and more into sort of routine applications, high throughput, as we all did, I think, 10, 15 years ago, both labeled and label-free. And then I sort of switched back into doing more bioinformatics, so doing more scientific workflows, automatic workflows, even automatically composing workflows for proteomics data analysis. So that's what I've been doing in the last 10 years or so, with some applications in biological species identifications, spectral libraries. And more recently, this last couple of years, I've also been, as many of the other panelists, fascinated by all these single molecules, potential next generation proteomics technologies. And since I don't know anything really about any particular technologies like pores or these DNA-based methods or Edman degradation methods, I try to model these things and try to understand what they have in common and what might be different, and how robust we might expect them to be, assuming that they will not be perfect right off the bat, but they will have some errors in their reads. And what does that mean for our ability to identify peptides and infer proteins like we do with mass spec data? So I just built some simulations, and I published a couple of papers on this that answered some of these questions to satisfy my own curiosity. And yeah, it convinced me that even if they aren't perfect, they can still be quite useful and I think robust. So some of the results were quite counterintuitive. So that was a great surprise. So yeah, I'm very honored to be part of this panel and I look forward to the discussions. And with that, I think I can hand it over to Mike.
Thanks, Magnus. So my name is Michael McCost. I'm at the University of Washington. I'm a professor of genome sciences. Next slide. So I want to kind of, my perspective as a mass spectrometrist is that mass spectrometry will remain the dominant proteomics methods for comprehensive proteomics. And I don't think people realize how hard proteomics is. So one of the reasons why methods like RNA-seq or spatially separating things in a flow cell work so well is there just isn't a lot of molecules. I mean, 400,000 mRNA molecules per cell means that you can get incredible coverage on a lot of counts, even from just 40 million reads. In contrast, there's over 4 billion protein molecules per cell. So to have the same coverage of proteins, and even if you had the same quality of data as you would have as a Illumina sequencer, would require 10,000 times more reads. Next. And if you consider practical limits like the spatial density of about, based on the fluorescence wavelength of 200 to 750 nanometers, you wouldn't be able to have a practical density much better than about 1 micron. Next. And this basically means that to get something like 10 billion reads would be about the minimum of where we're currently at with mass spectrometry, and LC-MS would take about a 10 centimeter flow cell. To get something that's more online with getting the coverage of what a transcriptomics is on the protein level, you need more like a flow cell on the size of about a meter. Now, of course, you can run many, many different flow cells, but that just gives you a sense. It's basically 10,000 times what you would get from so you would need to basically, currently, mass spectrometry measures and counts more molecules than Illumina does at about 1% the cost. Next slide. Next. Yeah, so and we're basically limited by very practical reasons. Next. Yeah, so one of the things that people don't realize is that we actually have achieved some incredible quality results by mass spectrometry, where basically we count single molecules. Instead of spatially counting them individually in a spatially resolved, we actually have a compression strategy. So instead of counting the same peptide 4 times, we have a measure of it once, and then we return 4 counts. It's the same thing the way JPEG compression works. You basically, it's a compression scheme. And in an LC-MS run, we basically have over about 500,000 ions that can be counted in about a second. And we can do this for billions of ions per hour and less than $100 per hour. Next. So again, just to kind of put this in a different way, so we normally think about current in the mass spectrometer on the left y-axis, shown in black, where we have a normal TIC. But of course, we can represent this as ions per spectrum by multiplying based on the amount of fill time. We can do simple corrections, of course, by altering based on the fill time to give us a better dynamic range and better quantitative results. So, so our current ions per second is basically analogous to doing like an RPKM or TPM normalization between flow cells. And currently in mass spectrometry, we can assign about anywhere from 15 to 50% of this current to a peptide sequence. So we're definitely measuring the equivalent of billions of counts per hour by mass spectrometry. Next. I think I'll kind of skip this, but we definitely have a lot of extra advantages. So one thing to kind of think about at LC-MS is that we're essentially doing tens of thousands of separate flow cells, which is basically just mass spectra, each with about 400,000 to 1 million ions on a millisecond timescale. We sort molecules. We also do an advantage. So imagine if you had an Illumina sequencer where it was 10,000 little mini flow cells, and in each of the flow cells, you put separate populations of the same molecule into each one so that abundant molecules didn't outweigh your count of lower abundance things. That's what we do by mass spectrometry now by using LC-MS. And we also use things like automatic gain control to basically extrapolate the signal and spend more time on lower abundant signals and less time on abundant signals. Next. Beyond the fact that mass spectrometry will play a major role, I think the future is going to be that there's a lot of proteoforms. Peptides don't always agree with the same protein. We have this habit of rolling them up into a single quantity, which often obscures the differences that occur between different samples. I also think there's a lot of work that needs to be done for improving the precision and dynamic range of data independent acquisition. I also would like to point where we're not always shooting from the hip and just running a sample. We actually have validated proteomics assays where we can basically say the limited detection, limited selectivity and specificity for every peptide and protein. And we actually have no missing data. We've also been working a fair amount to the point where we can get targeted proteomics that measure, thousands of peptides in a sub-hour with incredibly high sensitivity. And we can do this, that the the sensitivity can be, tens to so times greater than that of discovery proteomics. Next. And then, of course, we need to be able to get to the point where we can measure thousands of plasma proteins per hour from small volumes. We have lots of ideas on doing this. And I've really been a big fan of Ryan Kelly's work and others and in the single molecule space, a single cell space. Our interest is predominantly around trying to just characterize tissue heterogeneity and spatial proteomics. And I think I've gone over, so I'll hand this on. I believe Ryan's next.
All right, thank you, Mike. Yeah, so I'm Ryan Kelly. I'm focusing on single cell and spatial proteomics. And so especially single cell proteomics is receiving a huge amount of attention recently. There are a number of reviews and perspectives, such as this recent feature in Nature that kind of highlights the importance and the promise of single cell proteomics. I think the rapid growth in this area is especially remarkable, considering that the first publications that achieved, say, a coverage of at least a few hundred proteins in typical mammalian cells were just published in 2018. There we go. So some of the ways that we're now using single cell proteomics include comparing protein expression in individual motor neurons from ALS and control tissues to better understand disease progression and find potential therapeutic targets. We can also generate maps of protein expression across tissues to a depth of thousands of proteins and with a spatial resolution of 50 to 100 microns. So for single cell proteomics, the state of the art is really about a coverage of about 500 to 1500 protein groups per cell with a throughput of 10 to around 100 cells measured per day, depending on whether you're doing label-free or multiplexed analysis. And so in terms of coverage for the 5-year outlook, I think we'll be able to achieve a coverage of more like 5,000 protein groups per cell using things like ultra low flow separations, multidimensional separations, incorporating ion mobility, and with some improved data acquisition and analysis strategies that are under development now. In terms of throughput, I think especially with some of the global targeting modes that Chris and Mike mentioned, I think we'll be able to get to around 2,000 cells per day. This would be by multiplex analyses and with fast separations like 10-minute cycle times. Underlying all of this is, when we're going to the single cell space, is miniaturization. And I think it's pretty widely accepted that miniaturizing sample preparation using either the nanopots workflow or the proteochip or NPOP that Nikolai Slavov has recently reported is really critical for reducing dilution, for more efficient reactions, and to reduce non-specific adsorption. It's also just as critical to miniaturize the separation and ionization. So this on the right is just some recent data we collected over the holidays, comparing signal intensity in the top right for a 15 peptide mixture as a function of flow rate. And if we plot that, basically the signal per peptide input as we go from 2 nanoliters per minute to microliter per minute, you can see that in this hundreds of nanoliters per minute range where we're normally working, we're just seeing 3 to 7% of the signal that we see at these reduced flow rates per analyte molecule. So I think that really points to, we're probably not going to get to 2 nanoliters per minute anytime soon, but the motivation for reducing our flows and making our electrospray more efficient. And with that, I'll turn the time over to Miguel for the perspective of one of our favorite funding agencies.
Thank you. My name is Miguel Osandon, and well, something happened with my video, I think. I'm working the technology development branch in the Division of Cancer Treatment and Diagnosis at the NCI. So next slide, please. Yeah, I'm going to talk a little bit about the funding. As you are familiar with the funding at the NCI, funding favors by review very innovative projects, and that produce this project, when they're funded, they produce a lot of publication, very good publication in high impact journals. But when we analyze those projects and we see how many of these projects actually get to the clinic and actually help people, we found out that there are not many. So what happened is all of these technologies that we are funding at the NCI are getting stuck in the academic lab, don't go to translation. And one of the reasons is because sometimes the academics don't know how to translate the technology. So in this case, they just have all of this problem with the approvals, and as you see, it's very difficult. Next slide. So this is how we call the technology development pipeline. And when the pipeline advances in translation, the funding of the NCI decreases. So these are technology programs only, no other programs. And this is applied to proteomics. So we have several technology programs that target discovery, prototype, validation. And we have one technology program that target translation. So try to get the technologies in the academic lab go to the clinic. So they call the academic research partnership, and we have an R01, a U01. Now, when I do a search for applications, R01 application during 2019, 2021, we know that NCI receive around more than 6,000 applications per year. From those applications, when I did my counting, is around 300 applications average for the proteomic related projects in any type of proteomic, no any stage, particular stage. So as you see, NCI fund a lot of more application in other areas. But when you check in the last two columns, when you check the type of the success rate of this application in proportion, how many of the applications that they are getting are actually funded, you see is the same proportion. Basically, proteomic technologies as other technologies or other R01, they almost have the same rate of funding. That indicates that we are getting a number of applications in proportion funded corresponding to the number of applications that we are receiving. The problem is not that we are not getting applications. The problem is in which stage of those applications are getting funded at the NCI. And as you see in the technology development pipeline, right, the most of applications, they're going to come from the early development, prototype validation, and less application they're going to target, trying to actually move the technology, proteomic technology, to the clinic where actually it can help patients. So next slide. So we, in that vision, we try to target some programs, right? The Genomic Institute released this announcement, and we decide to participate. They have an R21 and R01, but we decide to participate only in the R01 for the same reason, right? We want the translation of the technology. We want more technologies, proteomic technologies in the clinic. Also, we have other announcement, and we can talk a little bit more if we have more time about this funding opportunities. And that's open to questions.
Okay, thank you very much. So, and sorry, I forgot to start my video when I was going. I've got a bunch of things to look at here. So maybe we'll stop sharing so we can see our participants a little bit better. And there we go. So I think we'll get these turned on. There we go. All right. So it is time for question and answer session. And so let's see what we have in here so far. Let's see, Jenny, this looks like a good one for you. What complementary platforms to mass spectrometers do we see coming to the forefront of proteomics?
So I was mentioning the screens that we have specifically for looking at receptomics, but I think we're really excited about some of these single molecule sequencing tools, which Chris would be better suited as the pair of us from Genentech to answer. They're very much in their infancy. You know, I think a lot of the things that Mike pointed out are very valid, that to be able to do exactly what we're doing with mass spec now, then it's a long way from getting to that point. But on the other hand, if we want to talk about highly sensitive analyses, and if we want to look at specific pathways to start with, or to just really be able to look at proteoisoforms and tackle that kind of question, which is still very important, then I think that these kind of tools are really going to make a huge splash at some point. But maybe I can defer to Chris on that, who's looked a lot more into this than me.
No, I think that was a good answer. I mean, like Jenny said, we know that they're early on in terms of where they're at, but I think we don't have to look at those, at least from our perspective, in terms of replacing everything we do with mass spec. I think what we want to do now is learn what the capabilities are and what questions might be better suited for that type of approach. And if the questions they can answer are really important to us, then I think that can be an exciting technology, even if it can't do everything we can do today by mass spec. And that's the way we're approaching it.
I was wondering if I could just add on a little bit of comment here. So, I mean, one thing, just great to get your guys' perspective on this, because this is something I definitely have not thought in complete detail on. But this idea of, you know, people getting, I mean, tens of billions of reads per sample. I mean, Illumina, that's, you're looking at like 10, 15 grand per sample, right? And so a protein-based assay is going to be less efficient than Illumina. So you're talking about substantially more cost than that. And there's just this, I mean, so I agree that simple mixtures may be the answer. So maybe really, but mass spectrometry is really well with simple mixtures in a few seconds of instrument time, right? So I think this is something that so I think there's this perspective, right? That proteomics, mass spectrometry, proteomics needs a problem to be solved because we haven't gotten the same coverage of things like we have gotten with RNA-seq on the transcriptome. The problem, what people don't point out as clear is that it would require 10,000 times more molecules that we're counting. That's just not possible with the wavelength of light. It's not even, it's like a theoretical limit, right? So I think that's just something I do want to kind of highlight here. That I think just needs to be said to people, so that people, when they realize and they see these technologies, nobody doubts. Let's say you get totally the same read quality as Illumina for proteins. Let's say you get better. You're still limited by the wavelength of light and the number of things that can be counted. And yeah, I just don't know how you get around that. So that's that fundamental flaw prior to starting.
So Javier, do you have any, so I think we, and thank you, Mike and Chris and Jenny. I think as a mass spectrometrist, I sort of think about single molecule, these emerging technologies as a group, you know, and I wonder if you could give us a little bit of granularity within that, as to say, which one of these technologies would you project to have the most immediate impact on the field?
So first of all, I think we have to be careful because mass spectrometry has like a 20-year head start, right? So, and these technologies, you know, like this is the first blip that these technologies are starting to emerge. You know, this movement has really picked up over the last 5 years. So where these technologies are going to go, we don't know yet. And I think it's a really exciting period that we're in because, you know, I guess the question that we have to answer for ourselves over the next 10 years in mass spectrometry-based proteomics is how far can we push it? And will we start stagnating in our ability to cover the proteome? That's what we have to answer. And we don't have a clear answer for that yet. And, you know, I think that if we start to feel like we're stagnating, then there is an open playing field of new technologies that are being developed in the background. And that's why I think it's so important to invest in these technologies now, because we, you know, like if that starts to happen, if we start to stagnate, then what will we back up on? So which technology is my favorite right now? I can't do that because they're you know,
I'm just trying to get you in trouble, you know.
They're all friends of mine. They're all amazing researchers.
Right.
They're all super enthusiastic, you know, and you know, I'm not going to point at one technology and say this is the one. And you know, the points that Mike is making right now on fluorescence technologies, you know, they're very likely very valid. But as alternatives to that, you know, there's nanopore technologies, and those can be parallelized. And you know, the future is bright, I think.
Yeah, it's good to be part of a field that's growing, at least.
Can I make a comment?
Yes, please, Miguel.
Yeah, I want to twist the question a little bit, which technology could be more useful, right? Because do we need to, I mean, I know the challenges, somebody pointed the difficulties, right, to the proteomic in relation to the genomic, right? But what type of question now we can answer if we can have that technology? And we need to have the whole extent of the technology. Do we really need to sequence the whole, like they did with the genomic, the whole proteome of the human, in order to answer some question that could be clinically useful? So I think the technology have to be moving, in my opinion, into the utility it's going to have, right? So mass spectrometry in the beginning, you know, was expensive. Now you have a lot of more mass spectrometers in the hospitals, right? You are going to be more useful, most useful for that. So the new technology that are coming, I would like to see technology that can be actually applied to answer some, in my case, clinical questions.
All right. So Miguel, while we have you on there's a question in the Q&A. It says, I think I know the answer to this, but it says, can NCI share the type of applications selected from the first round of single molecule sequencing grant funding?
We cannot share anything yet. The only thing that we can share is when the applications are funded, right? And no application can be funded yet. We can get all the information of the application in the NIH reporter, and I actually can submit the links for those to anybody related as after the application is funded. Before that, everything is confidential.
Yeah, and they also haven't been reviewed yet, so that's another part of it. So let's see. Maybe if I could direct this to Magnus. We'll get back to you, Javier. What role do panelists think will deep learning or artificial intelligence-based models play in the future of proteomics?
Yeah, that was actually also a question that I planned to ask the other panelists. I don't know if you want to bring up the slide that I sent you.
Sure.
Yeah, so I did a quick Web of Science literature search of the.
It will take just a quick sec to get there.
That's good. Okay. I can talk.
There we are.
Here we go. Yeah.
Okay.
So in the red bars here, we have the fraction of papers mentioning machine learning or artificial intelligence in proteomics in Web of Science over time, the last 22 years. And in blue, it's the same fraction than for papers in all of biology and medicine. And we can see that proteomics had an early start in using machine learning and deep learning, artificial intelligence, all those terms or topics appeared early. And then yeah, there might have been a brief trough of disillusionment around 2012, 2013 or something. And then sort of everything took off in 2018. And but what you see now in the last couple of years is that it sort of levels out. Okay, 2022 is very young still, but it looks like it's kind of flattening, whereas other fields are catching up. So that brings in the question, is there something holding back the application of deep learning in proteomics? Are we running out of good ideas, or what are the obstacles to applying deep learning? And if successful, then what are the most important contributions to the field that the other panelists see that deep learning or artificial intelligence more generally can make to the field? So I don't have the answers to these. I mean, I have some technical answers, reasons for why it's difficult to apply these techniques, but not the general.
So Magnus, I think one of your questions are really fantastic question. So that is, you know, why are we kind of feeling like we're plateauing? And I think one of the big challenges we have is trying to explain our data to machine learning and artificial intelligence experts. So right now, the thing that has been solved pretty well, or at least machine learning and artificial intelligence has contributed the most to, is the spectrum ID problem. They can wrap their head around it. They can grasp it. They can, I'm not trying to say like this, that they're not super intelligent. They are. They just have worked on different areas and different fields. I think we need to get better at explaining our data to people. And I think that's a huge limitation. And it'll continue to be a gap until we can do that.
All right, wonderful. Anybody else have anything to add on the general topic of machine learning, artificial intelligence here?
This is Miguel.
I definitely don't. So yeah, go ahead, Miguel.
Yeah, I think in the publication of something similar happened with the grant application that we have, in the sense that a lot of these machine learning methodologies off the shelf you apply to the data. So basically, the people that are developing this, a lot of them, they are looking for data, and genomic data and all other type of data are a lot more available, maybe easier. Maybe the reason is because, as Mike mentioned, it's so difficult to do proteomics, so it's difficult to get that data. And then the investigator decide to go to a low-hanging fruit and then apply the machine learning algorithm and tons of available data to get the publication in.
All right, thank you. So there's a question from Kerry Nugent. He asked, what would be the optimum LC column size, configuration, and flow rate for single cell proteomics? And I think that's a great question. There's not a clear answer yet, but generally what we see is smaller is better. So for example, Xiaorong Liu and Ying Zhu did a demonstration with open tubular liquid chromatography operated at sub nanoliter per minute flow rates. And they actually were able to see something, a number of proteins, even at say 1/200th the amount of protein that you have in a single cell. So going, that's definitely not practical yet, but I think it points to a direction we want to head moving forward. So that's a great question. Anybody else have anything to add there? All right.
One comment you didn't mention though, Ryan, was throughput, right? As you go to single cells, you're going to need to improve the throughput dramatically. So yes, we want to go to lower flow, but that's often counterintuitive or goes against the throughput increases that we need. So that's the solution that LC manufacturers need to help us solve, is how do we reduce the loading time through equilibration time while maintaining the robustness at low flow rates?
Absolutely. If you keep just like a normal sample loop and everything, all of those delay lines and tubing take forever if you go to the low flow rates. So yeah, absolutely. There's definitely more innovation needed there. So thank you for that. Let's see. Hossein, I'm not sure who to address this to, but Hossein Fazalinia asked, he said, thank you very much for this great webinar. One of the main challenges is proteomic analysis of undepleted plasma. Are MS-based methods able to compete with methods like SomaLogic? Anyone want to take a stab at that? Mike, I think you at least mentioned some plasma stuff in your spiel.
Yeah. So I mean, so the problem with SomaLogic is that it's an affinity reagent, right? And those affinity reagents, some will be really good and some will not. And depending on which portion of the protein they cover, they may give you different results than if they had a reagent to another portion of the protein. So they can get a very large number of data points. And one of the things that's a little bit perplexing currently is that some of the known important biomarkers don't show up in some of their experiments. And yet they find other things that nobody has seen before. So those, at some point in time, we're going to have to do a bunch of head-to-head experiments with SomaLogic and see which values are real and which ones are not, and which ones can be validated and which ones can't. But yeah, I think there's great, we definitely need affinity reagents, and using other types of affinity reagents other than antibodies is definitely powerful. So that's definitely something I just want to point out, is that just because you have an affinity reagent to a protein does not mean it's returning a quantity just for that protein. So that's one caveat.
Wonderful. How about our Genentech folks? Anything to add on some of these plasma
Yeah,
proteomics
I mean,
challenges?
again, it depends. It kind of depends on what question we're trying to answer as well. So if we know that if we did a depletion step and we're not actually losing the matter that we're interested in trying to monitor for biomarkers, for example, then we can work with plasma, no problem. But I think the problem is when we're going in blind and we don't know what we're looking for, so we don't know what we're losing because of some of these different reagents that are available for kind of normalizing the plasma. So I think, I know from our biomarker team, they're looking at various different methods in parallel to mass spectrometry because there are caveats to each of the different techniques. So it really depends on the very specific question they're trying to answer. But I think if you're looking globally, you know that you're going to incur some losses and have some biases regardless of which methodology you use at this point.
All right, thank you. So we have a question for Miguel from Sebastian Hayes. He asks, does end user experience have any impact on what technologies get all the way to commercialization? As in, the harder to use, the less likely to make it there. And you may be muted, yeah. There you go.
Yeah, in our experience, right, the end user experience could have some role, but it's mostly for funding, right? So the way that my challenge to you guys, to the investigator, is give me technologies that can be help patients in the clinic. That is my challenge to you, right? So you can do the whole sequencing of the proteomic, but if it doesn't help me to answer a clinical question, I don't know. Now, the incentive that I only can give in the NCI is through funding, right? So, and the problem is not the end user experience. The problem is where this funding is kind of easier to get, right? So, and it's a lot easier to start with a kind of a technology and a new technology. It's a lot easier to, in some way, to get it funded because, oh, wow, it's new, right? The technology is already proven, then, oh, it's something that I already knew that. So I'm not sure the user experience, as they stated, the question is going to play a much impact in this. It's more like, how can I use this technology to help someone?
Yeah, thank you. All right, so here's a question from Gil Omen. Excellent introductory statements and strategies. Do you have an emphasis on quality assurance for identifying uniquely mapping peptides, avoiding matches to sequence variants, or abundant proteins and isovaric PTMs? Are you interested in splice isoforms, things like that? So, so kind of a broader question is quality assurance, which I'm sure that some of us are very, very interested in. Anyway, anybody like to take a stab at that guy?
Well, I haven't got the answer to that, but one thing I do want to highlight that we're interested in looking at is the dark matter proteome. So we realize now that our search space isn't what we defined it as a few years ago. We know that there's non-canonical translation products. There's a lot of viruses that are actually just in the spectra that we're looking at. We now seen from the MHC world that there can be these spliced peptides and things like that. So it doesn't answer the question about quality assurance, but I think what we need to do as a community is to better understand what is actually in our proteome so that we're searching for all of the right things. And it's tricky to do. You don't want to explode your search space and suddenly have a whole bunch of false negative and positive results. But we've realized that if you look for some of these things, they do exist, and it could explain what a lot of the spectra are that we don't identify. In a typical triptych run, I think many of us can only identify a third to a half of the spectra in there, even if we put different post-translation modifications into place. So it's kind of a comment rather than an answer to this question, but I think it's an important one to just bring up as a community. How do we tackle that? How do we look for the dark matter that is out there?
Absolutely, yeah. You only see what you shine the light on, right? And so,
right.
Yeah, I would also add, we are very interested in not only splice isoforms, but even just homologous proteins or proteins that share a lot of sequence similarity. You start to see this when you start looking at some of the molecules that say there's a lot of degrader molecules out there, right, that will actually cause the degradation of a protein. Most of those occur in families of proteins that may share similar domains. And if you don't actually curate the peptides in the right way, you'll see that some of the peptides that are shared won't have the same quantitative result, basically, right? You won't see the same amount of degradation, or you might see a protein that looks like it's going away when it's really not. It's just a peptide shared with the protein that you're degrading. So those types of situations are when you might want to take a approach. I know Mike is pretty, excited about peptide-centric approach, but if you did that, you would actually see that the peptides that are shared have vastly different quantitation than the ones that are unique. So we are keenly aware of those, and it can make a big difference, especially when we're trying to, characterize a molecule and its activity, its on or off target activity.
All right, Javier, you have your hand up?
Yeah, so on the topic of the dark matter of the proteome, this is an area I've explored really deeply. And, what I find is, we try really hard to find all of these things. And maybe one of the biggest limitations that we have in exploring it is still the sequence coverage of the proteome. Because oftentimes, we search for mutations, for example, that we should be finding in cancer. We don't find those mutations, and it's not clear to me that we don't find those mutations because they're not there. It's confounded with our low sequence coverage of the proteome. Same with splice variants. Same with all sorts of strange things that we might want to find. We definitely find some, but probably not as many as we should be finding because of the current limitation in sequence coverage that we have.
Right, wonderful. So here's a single cell proteomics question. It's from Pankaj Dwivedi. Now we can detect 1,000 to 1,500 proteins in single cells, but how many of these are low abundance proteins like transcription factors, kinases, and receptors? Chris, you want to tackle that, or you want me to?
I can say it a little bit. So yeah, so I mean, at the moment, you're going to be detecting proteins mostly based on their abundance, right? So a lot of them would be abundant proteins. I think that doesn't mean that we can't find things that are interesting. I know Ying Zhu just had a paper out showing, he could identify cell surface markers that were unique to cells. And for some of those, they weren't found even when you looked at RNA-seq data and tried to kind of compare that between the different cell types. So even in the first 1,000 to 1,500 in one cell, you might be able to find some interesting proteins. If you look across all cells, which is a lot of times how you look at the RNA-seq data, you might be able to go a little bit deeper than that. But I think, part of where we're looking and kind of Ryan's 5-year outlook is to push that deeper, because being able to detect more proteins will help us to identify more of those transcription factors, kinases, other things that would be important for the mechanistic biology aspect that we're particularly interested in terms of like therapeutic activity. That will actually kind of open up another utility, right, of the single cell proteomics space.
All right.
So can I add one thing?
Yeah,
I want to
please.
point out, though, that this is not a unique problem to to proteomics, right? This is in the transcriptomic space too. There's a lot of zeros in the data, and and if you look just in yeast, where we know a lot, right? The average copies of a transcript for an essential gene is less than 1. Right. So there are not a lot of transcripts per cell, and in the population, these things are present. So we see them there. But when you start to look across single cells, there's a lot of ones and zeros. So I think I know we have this thing where we want to get a large number of thousands and thousands of proteins represented. But we just have to be, realistic in that that's holding things to a higher level than even a lot of transcriptomics data currently.
Okay, so we still have 15 open questions and about 5 minutes to go. So we're a little tight on time, but I'll try to let's try to get through as many of these as we can. So for Chris, from Aaron Jeffrey, he wants to ask whether the InSeq API is a proprietary tool or whether it might be open source or packaged with MaxQuant Live. So if you could answer that.
Yeah, so a lot of that depends on kind of the deals with Thermo. My take on this is that, packaging it up like MaxQuant Live is the most easiest thing to do, basically, because that's the model that's been done so far. if I can, I would like to at least have the portions of the code that users can edit, right, be open source, and then potentially a lower level that interacts with the API that's still provided packaged. But my hope would be for that to be open source at some point, but that all somewhat depends on the thermo and what they allow, basically.
Yeah, thank you. Question from Santosh Misal. Hi, Santosh. What is the prospect, you went away, prospect of single cell proteomics with respect to quantitative post-translational modifications? And that's a great question. And I think right now, basically, there are a number of groups working on that. I haven't seen much of substance come out in that area yet, but just bearing in mind that they're probably only present, even phosphorylation, maybe have 1% of the abundance of a unphosphorylated of the same peptide. So it's an even bigger challenge than single cell proteomics. So maybe we're catching our breath with doing single cell proteomics at all, but I think in the next few years we'll also see that's clearly another frontier in this space, is extending these measurements to single cell or to PTMs. Okay. Here's one from Benjamin Neely. It seems pore-based techniques have more upside based on ease of use and portability, even if worse performing. Could non-MS based techniques take off for these reasons alone? What do you think, Javier?
Sure. I think that portability it has been really important for DNA sequencing. I could see something like that in the far future. These technologies aren't ready to do that yet, but they strive to be. Last comment. I had one more comment. I've lost my train of thought. Go ahead.
Okay. Mark Wingard asks, what do you think of the potential of smart platforms enabled by things like MaxQuant Live? Any takers there?
I think they could have a really important place going forward. I mean, I think some of the global targeting stuff that has been done with MaxQuant Live, some of the stuff Mike has done, I think that could be really useful in terms of single cell proteomics training, right? Those types of applications. So I think going forward, we'll see maybe the vendors will start, kind of incorporating more of these things too. It won't just be the users, but a lot of the innovation might be gone first by people out in the field doing these kind of more innovative things and then get adopted later.
Yeah, absolutely. I think now, having worked in this space for a while and then looking at data dependent acquisition, it's hard to come up with a more inefficient way of measuring the proteome of single cells when you think about how long you need to accumulate each ion and what's happening to all the other ions that are co-eluting. They're just getting wasted, right? So smarter strategies there. I think there's plenty of room, and I think some of these kind of flash talks have made that point. There's plenty of room to do things better than we're currently doing, and it's a wide open space here. So anyway, well, we are right at the hour. Should we turn the time back over to Olga or anybody else? Or final thoughts from anyone?
Oh, well, thank you very much. I just would like to thank everybody on behalf of the US HUPO. These conversations came out of Twitter threads, which we have seen in the past. And we hope to continue, bring the Twitter discussions live to the entire US HUPO community. And we're also happy to do other things. So if anybody in the audience has suggestions of other topics or other types of panels, or any other activities that we can do while being on Zoom and being somewhat isolated from each other, and we can still have these conversations, please let us know. We will be most happy to continue.
Can I comment on one thing that Olga just said? I think one thing that is important is if people don't follow threads on Twitter, there is a lot of these really good discussions occurring. And also, if people don't know me, I tend to be kind of playing devil's advocate a lot too. So people often, my students definitely know that, my trainees, but I think it really is important to get these discussions out there and to kind of think critically about them. It's not me being critical or anything. It's just me trying to apply the other perspective. But I do think, though, that is a great mechanism for people to have some of these discussions in a fairly limited, finite, 140 characters or so.
And then for some of these things, we take them up and have a broader discussion live. And so this is very, very valuable. So thank you very much for joining us today. Thank you to the audience also for their participation. And please stay in touch with US HUPO and please follow the news, and the conference is coming up. So I hope it will be possible for you to join us there. Thank you very much.
All right.